Hello, everyone. Thanks for the organizer to give me the opportunity to talk about my recent work on the relationship between gene expression and the agronomic traits in polyploid wheat. I'm Fei He from the Institute of Genetics and Developmental Biology, Chinese Academy of Sciences. First, I'll talk about some background. Compared to arabidopsis, wheat has a huge genome. It has 3 highly similar subgenomes with more than 80% of transposable elements. The evolutionary history of wheat genome is pretty complicated. The 3 subgenomes share part of their genes inherited from the same ancestor, and those genes are called Homoeologs. Since there are 3 highly similar Homoeologs copies in the wheat genome, the study of the expression becomes more complicated. As a crop, wheat can be grown in many environments. It is the most widely grown crops in the world. While the genotypes of different wheat lines have been explored by many studies, the expression diversity have not yet been explored so far. So in our study, we assembled a panel of 200 wheat lines collected from different parts of the world. Those wheat lines are grown together, and the 2-week seedlings are used for RNA-seq. We used multiple technologies to obtain genotypes, which includes about 2 million SNPs. We have 50,000 transcripts detected and expressed in this study. The association tests were performed between those 2 million SNPs and those expression data we followed the published pipeline to perform this association analysis. We controlled the population structure, the confounding factors, and we also did the normalized the expression data properly. Here is a calculated eQTL map. The most obvious pattern on this map is a diagonal line representing a strong signal of cis eQTL. There are several other interesting patterns on this plot, such as a small diagonal line formed between Homoeologs regions. The series of blocks here, as you can see, are a big translocation from distant relatives. The genes on this translocation are tightly linked, and they are co-regulated by many different SNPs. The position of the eQTL relative to this target has not expanded a lot compared with maize and other species. And the eQTL tend to locate on both sides of the gene. And so we do find that the smaller D genome has a longer distance between the lead eQTL and the transcriptional start site, probably due to the reduced diversity of the D genome and the longer ALD. Power law is a typical feature of network. The eQTL map is in fact a network between SNP and the gene. So if we check the distribution of the target for gene, we can see that some genes are regulated by, we can observe that there are some SNPs regulate a lot of genes, and some genes are regulated by a lot of loci. For some of those so-called regulatory hotspots, we do observe gene ontology enrichment among its targets, suggesting co-regulation of genes involved in the same pathway. Another pattern from the eQTL map is the regulation between synthetic genomic regions. Those regions have similar gene content. It looks like that cis element of one copy of the Homoeolog also regulates its Homoeolog's copy on the other subgenome. Since the synthetic regions of the wheat genome might be in very close contact in a 3D space, we're wondering if those trans eQTL are in fact cis eQTL via 3D commenting contact. So we checked the published HiC data, and we do find that the HiC singular and the eQTL singular between those regions are positively correlated. Here's an example showing how the trans effects is in fact a cis effect via 3D contact. Those are the two copies of the same gene located on the two different subgenomes. The sleep highlighted here in red color is eQTL for both copies of the gene. It is a cis eQTL of the bigenome copy and the trans eQTL for the digenome copy. The HiC data shows that those two genomic regions are in strong contact. Which means that this so-called trans eQTL is in fact a cis eQTL in a via 3D contact. We also perform typical eQTL analysis by comparing the overlap with functional notations, and I'll skip this to save some time. We observed enrichment of cis eQTL in GWAS heat for most traits we phenotyped for our panel. This means that we can identify better identify gene candidates using eQTL data. So we used the well-established method. Here we used the SMR method to integrate the eQTL singular and the GWAS results to predict the gene candidates, and we predicted about 300 gene candidates. Since there are relatively few genes cloned in wheat, we used co-expression and the infers regulation to build up a gene network in order to find out some clues between those predictions and the agronomic traits. Interestingly, gene candidates of the same gene candidates here of the same traits tend to be co-expressed, suggesting genes within the same pathway were predicted as the candidates of the same trait. As you can see here, most homologous copies tend to be co-regulated. About 75% of the pairs, of the homologous pairs, showing positive correlation, and only 1% of pairs showing negative correlation or dysregulation, as we called it in our study. The eQTL map is mostly loci with strong effect on expression. In order to have a comprehensive evaluation of genetic control of gene expression, we calculated a sleep-based heritability. We partitioned the expression variance onto 3 subgenomes, and we found that the A and B genomes have more control of their own gene, while the D genome is more controlled by the other 2 genomes. This is probably due to the bottleneck and gene flow from tetraploid ancestors, where the D genome diversity is heavily reduced compared with A and B genome. We noticed that when we analyze the sleep diversity and the gene dysregulation, with the accumulation of cis regulation, the balanced or coordinated expression between homologs are disrupted. As shown here, the x-axis represents a correlation between a pair of homologous genes. The y-axis represents the number of sleeps located in the 1 megabase flagging sequences of the gene. When we looked into the actual expression pattern of those negative correlation, we found that they are caused by low expression or gene silencing in a subset of the weight lines. In our panel, there are 59 genes showing this pattern, and we define them as dysregulated genes using some standards. Here we use TPM-2, the TPM mean minus standard deviation of the population in the population. Basically, we are looking for the lowly expressed genes or the silenced genes compared with their homologous copies. So we find that with the accumulation of dysregulated genes, the weight tend to have larger grain weight and a smaller number of spikelets per spike. Compared with randomly selected genes, dysregulated genes show a stronger correlation with a list of agronomic traits, such as here grain weights, grain area, grain weight, and also the accumulation of dysregulated genes is negatively correlated with plant height, spike length, heading date, and spike number. So we performed genomic prediction using the expression data of those 59 genes. So this is a expression profile of those genes, and here is the agronomic traits phenotype in our for our 200 weight lines. Here is a number of dysregulated genes in each weight line. When we correlate, when we use expression data of those 59 dysregulated genes to perform genomic prediction, we observe the accuracy. The accuracy actually is pretty good. So you can see the predicted trait value versus the actual trait value is the correlation is 0.35 compared to what's reported in maize. So this is a pretty high correlation or pretty high genomic prediction accuracy. This means that those 59 genes can predict traits very well, despite a lack of direct correlation between their expression profile and the traits. So we cannot find any direct evidence of the association between each of those genes and the agronomic traits. Although we do not know why those dysregulated genes impact agronomic traits, we observe elevated linkage and distribution between the corresponding homologous genomic regions compared to the genomic background. This means combinations of analytic pairs of homologous genes are under selection. Different breeding programs might choose to either accumulate or purge out combinations of those analytic pairs in order to achieve different breeding goals. So that's all for my talk. And this work was mostly done at Kansas State University when I was a postdoc with Edward Akerlof. And now I have my own lab at the Institute of Genetics and Developmental Biology at Chinese Academy of Sciences. And this work was funded by Bill and Melinda Gates Foundation. I want to thanks for your attention.